This week, our subject matter is scientific technology and how it relates to human nature and to scientific knowledge. And in particular, we're talking about CRISPR technology and MRI technology. The theme for this week, and again, this week we'll be letting the individual YouTube videos and the readings describe the details of how MRI works and how CRISPR works to you. I'll be just highlighting the philosophical points and morals to get from those stories. And the big moral of the story for this week is by their deeds shall you know them, which of course is a quote from the Bible, which could be taken as the motto for our subject matter for this week, which is functionalism, which is the scientific theory that says by how a thing functions, will you know it. If it looks like a duck and it acts like a duck and it quacks like a duck, then it's a duck. So technology tends to move towards functionalism, since it's aiming at manipulation and control of how a thing functions and not the knowledge of a thing. So modern technology is going to find ways of overcoming some of the limits of scientific knowledge. Scientific knowledge runs into limits, as we've seen in quantum mechanics and other subjects. In the complexity of the objects and the mechanisms and causes that it has to understand. So technology finds ways of controlling a thing without understanding all of the details of how and what makes that thing work. So if you're a car mechanic, you can understand how to fix a car without understanding the technical specifications of every part. You just know what each part does, and then you know how to order it, and you just replace the parts instead of rebuilding them from the beginning or understanding exactly how all of them were manufactured. So that's the view called functionalism. Functionalism sees things in terms of how they function rather than their underlying natures. It's ironic that the origin of functionalism was in the philosopher Aristotle, who came up with the idea of essentialism. Remember, essentialism is the view that all objects have natures or essences, and that determines their purpose or their function. Aristotle came up with the word for which we use to describe the functional units of the human body, or organs. Organ in Greek just meant tool. So Aristotle thought that you would know an object and name it by the function that it performed, precisely because he thought that the function that it performed was part of its very nature, part of its very essence, part of the purpose to which it strove. And that was part of the natural law theory that said the morality of an object is determined by the nature and the particular function that it played in the universe as designed by God. We're going to be looking at a different form of functionalism called mechanism, which views everything as a machine. A machine is precisely an object that's completely defined by the functional role of its parts. Think of the engine of a car again. We don't really need to know what the parts are made of. They could be made out of plastic or wood or any type of metal, as long as the specifications of those objects allow them to function in the right way. And what makes the engine run is simply the formal relationships of those parts and how the gears and pistons and all those things fit together. There's no soul, there's no mind, there's no internal nature that's guiding the machine. The machine simply functions by the arrangement of its parts, and its parts are not defined by the material that they're made of. They're defined merely by how they can function or operate in their specified role within the machine. So we're going to be looking at how in CRISPR and MRI human nature is conceived of as a machine in virtue of the technology, and how the limits of that technology bump up against some of the ways in which scientific knowledge has limits. So in MRIs, we found a way of overcoming the complexity of the human body to understand what's going on in the body and in the brain. As you read in the reading and in the YouTube videos, MRIs detect merely the orientation of the spin of hydrogen nuclei in water molecules. And they're deriving lots of complicated information about the structure of organs and the structure of the human body from that very indirect route of measuring certain obscure properties of the components of some of the water molecules that are in our body. fMRI, which we'll be talking mostly about, you may use MRIs very often to picture organs for their medical uses. But we're going to be talking mainly about fMRI, which detects the flow of blood by seeing differences again in the water that makes up oxygenated blood and deoxygenated blood and their reaction to magnetic fields. That will show us how blood is flowing in and out of the brain over time. Not only is the route by which we know the human body very indirect through just these obscure properties of the water molecules, but the results are very complicated. It's not as if you can just look and see what the organ is. The images that you see on an MRI screen are actually constructed pixel by pixel by looking at the results for each of those regions in space and having a computer calculate very complicated statistical models to determine when to show it lit up, or how to gradate the color or the vibrancy or the light of each of those pixels that constructs the images that you see. And those statistical models have some of the same defects that we talked about in statistical models last week. There have been recent studies that have shown that the models that were used in computers for fMRI from many studies over the last 20 years or so had been mistaken, and that many of the results from the last 20 years may be called into question because of those mistaken statistical models. Although those would have been the types of MRIs used in research, not the type that were used in hospitals. So we're going to talk mainly about how fMRIs are used to talk about the brain. There's been sort of a revolution in how we understand the human mind and human consciousness in virtue of our newfound ability to look inside the brain and see exactly what parts of the brain are being lit up. Again, it's useful to recognize just how complex the structure of the brain is, and just how indirect and simplified the models that we're seeing through an fMRI are. But here's a simple example where we can see the regions that are active in a person's brain when he's shown a picture of his wife. The visual center of the brain is lit up, and then the frontal lobes, the part where his thoughts and memories and emotional associations with his wife would be located. So the first time, this marks a hallmark in the scientific study of human beings, because for the first time we can look inside the human brain and say, this is an emotion, this is a thought. That view, remember, was called reductionism, or reductionistic mechanism, that said that all things can be reduced to their parts and the functional roles that they play. So here's an example of someone who's asked to remember scenes and then to ignore them. There's some differences in how young adults and older adults see it. But when you're asked to remember a scene, certain portions of the brain light up, and when you're asked to ignore the scene, the same ones light up, but you are actively suppressing them. You've seen images such as this one, where you can tell the effects of various different drugs. So here's the dopamine level of a brain without cocaine, and when it's on cocaine, the pleasure centers of the brain are activated by the drug, producing the effect that we describe as the excitation produced by the drug. We can peer into the brain in ways that we couldn't have before. So here you can see that the parts in red detect when someone is lying, We now have lie detectors that can look at the galvanic skin responses and the movement of the pupils of the eyes, and things like that. But now we have a way of seeing when regions of the brain that would be associated with fabrication would be activated, apart from the normal parts of the brain that would be activated in language or imagining the scene we're asked to describe. So here's a study that shows a real orgasm on the right versus a fake one on the left. And in the fake one, the part of the brain associated with fabricating the emotions and actions that you're faking would also be activating. Here's another one that you're familiar with. On the right, you see a severely depressed person rather than a normal one, and the levels of serotonin, another neurotransmitter, are shown to be depressed or reduced from a normal person. Many of the drugs that now operate on depression cause an increase in our serotonin levels artificially. They're called selective serotonin reuptake inhibitors because they inhibit the process by which serotonin is degraded in the brain and produces a higher level of it. So now we can understand even emotional states such as depression or happiness or elation in terms of the level of serotonin in the brain as detected by these MRI or PET scans. Here's a case where we can see what pain is in someone's brain. When they're in pain, these pain centers of the brain light up. And in cases where people are looking at a picture of someone in pain and feeling empathy with it, the same portions of the brain are lit up. And here's a case where someone who is diagnosed as being psychopathic, when they look at pictures of people in pain, whereas a normal person has the pain centers of their brain lighting up in empathy, the pathological person or the psychopath or the sociopath doesn't feel pain for other people. Here's a case where we can reduce a very complex social deficiency to a particular feature of the brain and perhaps figure out a way of altering it or remedying it by remedying certain physical features of the brain. This is an fMRI picture of the particular activation of someone who is love-struck or who is in love. So we can even take some of the most fundamental human feelings and reduce them to the activation of a particular region of the brain. And in this case, being in love activates many of the same pleasure centers that are associated with drug addiction. Even the most religious and metaphysical of human mental states can be studied in this way. Here we have the picture of a Buddhist meditation with the activity of his parietal lobe shown to be decreased, in some way calmed by the meditation. The details of these aren't important, rather the general idea. So the technology allows us through this very indirect route to see very clearly what functional changes in the brain are associated with feelings and emotions. Reductionism or physicalism or materialism, which you see on the bottom that we've already studied, would say that if you're going to separate the relative and the absolute, just like what we do for red, we would say that redness is our subjective way of perceiving the wavelengths of light. If we separate the relative from the absolute, we'll see that redness really is merely the wavelengths of light. The feeling or sensation we get is just our subjective way of perceiving it. It's not really out there in the world. When we hear a tree fall in the forest and we hear that sound, what we're hearing is the effect that it produces in our brain. And the actuality are the wavelengths of the air vibrations that are caused by the falling tree. So when the tree falls in the forest and there's no one around to hear it, it doesn't make that change in our brain. It doesn't make that special feeling. Instead, it only makes the vibrations of air. So reductionism or materialism would say that we have to separate the relative and the absolute completely, and that the real thing is only what exists in the world apart from us. So now with fMRIs, for the first time, we can apply that method of separating the relative and the absolute to our own feelings and our own emotions, because we can see from the outside, someone who's not experienced them, we can see what's going on in their brain when they see red, or when they're in love, or when they're remembering something, or when they're on cocaine. And we can say that no matter how it feels to them, the reality of that, the scientific reality, is just that their love, their pain, is that state of the brain. The reading that you had by Frank Jackson about Mary the neurosurgeon is an argument against that, that points to what some people think are a deficiency in the scientific method, in separating the relative from the absolute so unambiguously. They ignore a feature of reality that can only be perceived from the inside. This view is called non-reductionism, or property dualism. The famous example is that Mary is supposed to be a neurosurgeon who's grown up in a black and white room where she's never seen any red object. So she knows everything there is to know about what goes on in people's brains when they see the color red. She's seen all of the fMRI scans, and she knows exactly what the objective reality of redness in the world is, the wavelengths of light. She knows exactly what the objective reality of people's perceptions or feelings of red are by looking at the fMRIs of their brain. Yet Jackson argues that there's still something that Mary doesn't know about redness. She doesn't know what it feels like to actually perceive redness. She doesn't know what it feels like to actually be in love if all she's seen are the MRI scans of people like the one I showed you, of people who are in love. So property dualism says that mental states and physical states are different sets of information, and neither one can be eliminated if you left out how it feels to be in love, or what the subjective nature of the human experience was. You'd be leaving out an important part of reality, which science, by its very nature, since it has to separate the relative and the absolute, will leave out. That was the meaning of some of the famous romantic poets' claims that you heard before, that reason misshapes the beauteous forms of things. We murder to dissect. That was William Wordsworth's words. He thought that science, and separating the relative from the absolute, dissected or murdered or took all the life and the feeling out of things just by using the scientific method, but that the life and the feeling of things was an essential part of reality, even if science couldn't see it. The other thing to recognize about MRIs and about most scientific technology is that they're seeing things from a great distance, and they're seeing things in terms of their functional properties rather than seeing them in terms of the real natures of those things. So we said that one of the limits of science is the complexity of the objects they're studying, even though we think it's true that every particle in the world follows the laws of physics and is completely determined by them. It's far too complex when we try to predict the weather, far too complex to measure every particle of air and its velocity and momentum and all those things. So we try to simplify it in terms of cold fronts and weather patterns that talk about the functional behavior of certain large and complex configurations of weather. In the case of the human brain, we've hit upon a unique limit to science because in this case, the processor that's trying to understand and the thing that's trying to be understood are precisely the same. The human brain has trillions upon trillions of neurons, which means that it's immensely complex. And of course, the thing that's trying to understand the human brain, which is of course our human brain, is limited by those precise number of trillions of neurons. So it's literally impossible for a human brain to understand a human brain in all of its complexity, to complete the scientific story and reduce everything to its real necessary causes and eliminate completely the naked face of chance. So instead, most technology, as we said, simplifies greatly, latches on to something that they can functionally observe, and tries to identify the object with those functional regularities that they can observe and control and manipulate. So an fMRI image that tells us about the functioning of our mind, remember, by detecting subtle alignments of the magnetic spin of hydrogen nuclei in water of the various different parts in our body over various different periods of time, and then interpreted by complicated computer statistical algorithms. That is an indirect view of what's really going on in the complex system of trillions of neurons of our brain, in the same way that viewing what was going on at the World Series by taking a satellite image of the city and seeing things flowing in and out of this large stadium and seeing the cars move in and out and maybe even tracking, the energy flow of the electricity flowing from the power plant. We'd know that something was going on there, and we'd know what it was, but it would be silly to think we know what's going on in the baseball game by describing it merely in terms of the flow of things in and out of the stadium as viewed by the satellite. So the same could be said for our attempt to know what's going on in the human mind according to fMRI. Two types of limitations to pay attention to there. One is limitations of knowledge. Our brain is too complex, so an fMRI or technology inevitably simplifies and looks at functions instead of the real mechanisms or causes of things. And secondly, separating the relative from the absolute seems to eliminate the relative, which for human beings may be precisely the parts of human experience that we think are most beautiful and most valuable. What it feels like to perceive red, what it feels like to be in love. So functionalism is seeing an object as a black box. We don't care what's on the inside of it. We don't care what it's feeling. In the case of the human mind, we can treat a human being as a black box. But it's also not even caring about what material it's made of. Remember, we said that we don't care what the engine is made of, whether it's machine or metal. So the fMRI doesn't care what's actually going on in the neurons. All it's doing is tracking how much water is flowing in and out of it, or the structure of the water in the membranes of the thing. It doesn't know what those membranes are doing or why they're doing it or how they're related to the other one. It treats them as a black box and only can compute the pattern of inputs and then outputs. You tell the person something, they see something, and then out of the MRI will shoot a screen that has pixels that are lit up or not lit up, showing a shape of activation. So in functionalism, a thing just is its program, its software. Functionalism is often associated with the idea that it's software that matters rather than hardware. Note that the same software can work on different hardwares. You can have the same pattern of inputs and outputs. So your Microsoft Word or your email program may work exactly the same on different physical computers or on an Apple or a Windows PC. The functions are said to be substrate neutral. It doesn't matter what material they're working on. The matter literally doesn't matter. All that matters is that they function the same way, that they somehow instantiate the same software, the same program. So as we said, functionalism was a way of overcoming the complexity of nature and the defects of materialism. Materialism is too complex. The world is too complex to break everything down to its smallest parts. And the world is too complex to compute all of the relationships between all of the particles. So instead, we identify larger level abstractions. So economics or biology or sociology now deal with functional units that are like the flow of capital or interest rates or the behavior of crowds or the evolution of species. Those types of sciences, biological and social sciences, would never have been possible if everything had to be reduced to physics or to chemistry. The world is just too complex. So instead, we identify functional units, letting the chemistry and the material natures of the things be a black box as long as we can understand the patterns of inputs and outputs. A good example of that is how we understand genes in their relationship to the human body. Because there's no such thing as a gene in nature. What exists in nature are complex strings of chemicals, complex strings of DNA molecules. A gene is a functional unit. It's something that does something or produces some trait in the organism, and by their deeds you shall know them. We identify genes not through their physical structure, but by the traits that they produce in the living organism. CRISPR, the new technology that's getting so much news, which is an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, which refers to the repeating pattern of base pairs in the strand of DNA that allows the CRISPR editor to find and cut a DNA at a particular place. The CRISPR technology is a mechanical means for editing functional units of DNA molecules that's based upon a purely functional and software-based mechanism. So CRISPR was actually an ancient bacterial immune system that edited the genes of living cells. The bacteria would be invaded by a bacteriophage or a virus, and it was very important then for them to be able to incorporate a little bit of that virus's DNA into the code of the bacteria, so that then they could produce antigens which would match up with their DNA perfectly, and then latch onto it and destroy the virus DNA. So this mechanism for inserting genes into a DNA strand and cutting DNA and inserting genes into it became the basis for this new technology called the CRISPR technology. Again, you'll look at the details of it when you do the reading or watch the YouTube videos. And again, the details aren't that important to us. You should get the basic idea, though, of what's going on. That it's got a target or a guide RNA which finds the correct place to cut the DNA by matching, finding a match with a certain set of spacers or base pairs in the DNA. And then there's the Cas9 protein, which cuts the DNA after it's been matched, been guided to the correct place. And then after the DNA is cut, the repair mechanism can then, if it wants to, insert new genes or new base pairs that have been prepared and inserted onto the Cas9 module to insert the new gene into the DNA. For our purposes, though, the big picture is more important than these details. And obviously, just as from looking at these diagrams and hearing my short description of it, you can see that this is a mechanism. It's like a machine. We don't care. We haven't even talked about what actually all these things are made of and what they look like on the molecular level. We're seeing them as functional units. And in fact, there are different versions of this based upon different functional units. In the actual bacteria, there was a Cas1 and a Cas2, two different actual molecules that perform the function of the Cas module. And either one of them would give immunity. They didn't need both of them. If you eliminated one, it would still work because it doesn't matter what the material it's made of is, or whether it's Cas1 or Cas2, as long as it can play that same functional role. That's all this mechanism cares about. And DNA is a marvelously complicated functional mechanism for reproducing organisms. And it's important to recognize when we're understanding the process of editing genes in somatic cells, which are cells that won't reproduce or be passed on to the next generation, or in germlines, where the changes we make will alter the human genome forever and be passed on to any children that who have that therapy. It's important to remember what the relationship between genes and organisms are. Genes do not make an organism, do not make a living thing. Genes are instructions for the production of proteins. If you're unfamiliar with that, you probably studied this process many times, but you may want to click on this and watch the little film strip for 2 minutes that will remind you of the process by which DNA and the regions of it, particular regions of it, which are genes, are the instructions to produce proteins. In the cell. And then each of those proteins then fits together geometrically, like the geometrical constructions we studied earlier in the semester, to create a certain shape of cells which produce a certain type of organ, which when put together produce the human body. So this is an extreme version of reductionism and of mechanism. The gene is just a functional unit to produce a certain protein, and yet somehow we speak of the gene as producing blue eyes, or of being for aggression, or of brown hair, or of for a certain type of body type. Yet there is no gene that determines how all these things will be put together. It's merely the geometrical shape and structure of the proteins being made in a certain order at certain times that causes them to coagulate into the shapes of cells that produce the various different organs and the wonderfully complex organism, which is a living thing. So this is a case where the functionalism and reductionism in genetics tends to make us ignore, again, the things that are about the reality of an organism that are not merely functions. It ignores the immense complexity. So even though two twins may have exactly the same genes, we all know that they'll come out to be different. A person isn't just merely their genes, because the individual matter that they're made of, sometimes it does matter. Things aren't black boxes, and the immense complexity of a human organism will make mistakes and details and complexities of the matter and the situation in the womb and their development of the organism will change radically the actual structure of the living organisms. So sometimes the little parts of things reassert themselves by interfering with the functional working of the genes, in the same way that sometimes your computer will break down, or uniqueness in the setup of your particular hardware will cause the software to act differently. And probably more importantly, the genes just produce the proteins, how they function together in organs, and how those organs come together to make a human body, and how those bodies come together to create a human society, are determined by the genes only in the most specious of levels. The genes create the parts, but how those parts interact isn't something that's coded for in the genes. So oftentimes this functionalism causes us to overlook the way in which the object works as a whole. So our two big takeaways from our brief look at MRI and CRISPR should be this. First, that both MRI and CRISPR work by overcoming the limits of scientific method. They treat objects as functional units, as black boxes whose natures are really unknown, but whose activity can be observed and controlled. So just like quantum mechanics, it was a way of overcoming our failure to get at the real natures of things and to limit the naked face of chance. In this case, where we can't understand the vast complexity of things, we simplify and get at things through their interactions with water, as in MRI, or through their functional units, or genes, as in CRISPR. And second of all, science works through reductionism, by breaking things down into the functional units. In this new form that we identified as mechanism, or functionalism, it ignores how things work as a whole, because sometimes it isn't the parts that matter, it's how those parts are related and how new properties arise in their interaction and their relationship. So genetics will ignore ecology, for example, and how a thing functions in its environment and its complexity. And it will also ignore how a thing relates to the subjective nature of human experience. And that'll have important moral implications, because when we ask, should we base our understanding of human behavior on the MRI, or probably more directly, should we use CRISPR to alter the human genome, there will be two different types of objections based upon these two different limitations. On the one hand, someone might say we shouldn't use CRISPR because we don't have yet enough knowledge of how the emergent properties will affect each other, just as some people object to GMOs, because even though we know what the genes are producing, how genes interact in an organism and then how the organism operates in its ecosystem are far too complicated for us to understand, so that we don't know whether there'll be unintended consequences of allowing genetically modified crops into the fields. So in the same way, even though we may know what this one gene will do and how it will produce a protein, we don't yet understand how it will function in the complex whole, which is a human organism, or in the complex whole, which is the ecosystem in which the diseases of a human organism operate. And second of all, and we'll talk about this more in our next week's discussions, the mechanism of modern technology tends to eliminate the subjective nature of human experience, how a thing feels to us, and most often the moral content of any situation is determined by how a thing subjectively affects human experience, how it makes human beings feel. So we might also argue that modern technology tends to dehumanize or take the human element out of the natural world, and therefore some of its applications can lead to acts of great immorality. That'll be our subject matter next week when we look at Henrietta Lacks's story and the story of HeLa cells.